Right, so you've spent this chapter mastering the standard classification systems. You literally just nailed down the fundamental rules that govern the domains of archaea and bacteria. You know, their shapes, their standard cell walls, and you've got their typical metabolic pathways down cold. But before we officially close the book on this topic, we really need a grand finale. Today's explainer is all about the exceptions. We are looking at the prokaryotic rule breakers. It's a huge question, right? What exactly happens when prokaryotes completely defy standard bacterial norms? Well, these microbial rebels have evolved some absolutely astonishing workarounds to survive, and unfortunately, in some cases, to infect. We need to understand these outliers on a really deep level, mainly because right after this, we're moving on to an entirely new chapter on eukaryotic cells and microorganisms. So to understand exactly where we are going, we first have to master the microbes that blur the lines. Here's our roadmap for today. As future healthcare professionals and microbiologists, you'll need to master 1. Environmental rule breakers, 2. Clinical rule breakers, 3. The extreme domain, and finally, 4. The 3-domain matrix. All right, let's kick things off with Section 1, Environmental Rule Breakers, Photosynthetic Prokaryotes. Okay, so we're talking about bacteria that have evolved highly surprising metabolic mechanisms just to harvest light. Cyanobacteria, you might know them historically as blue-green algae, are absolute powerhouses. They execute oxygenic photosynthesis, which means they actually utilize water as an electron donor, splitting it to release oxygen gas right into the atmosphere. To pull this off, they use chlorophyll A, pretty much exactly like plants do, along with accessory pigments that give them that signature blue-green color. But structurally, they completely break the standard prokaryotic mold. They've got these extensive internal photosynthetic membranes called thylakoids to house all their light-harvesting machinery. They even feature these specialized, thick-walled cells called heterocysts, which are dedicated exclusively to anaerobic nitrogen fixation. Talk about overachievers. Now take a look at the contrast when we literally just remove oxygen from the equation. Enter the green and purple sulfur bacteria. These microbes execute anoxygenic photosynthesis, meaning they don't produce oxygen at all. None. Instead of water, they use reduced sulfur compounds like hydrogen sulfide as their electron donors. They use these highly specialized Bacteriochlorophyll pigments to absorb longer infrared wavelengths way down in deep, dark anaerobic aquatic sediments. But here is the absolutely key structural divergence you need to remember. When they process that hydrogen sulfide, purple sulfur bacteria deposit pure sulfur granules internally, right inside their cytoplasm. Green sulfur bacteria, on the other hand, deposit those same sulfur granules externally, completely outside their cell walls. Moving on to Section 2. Clinical rule breakers, obligate intracellular pathogens. For those of you heading straight into healthcare, you'll want to lean in for this one, because the pathogenic impact of these specific microbes is massive. It's an absolute game changer. The crucial point here is wrapping your head around this definition. The obligate intracellular lifestyle means these are bacterial pathogens entirely incapable of multiplying, or frankly, even surviving outside of a living host cell. Why? Well, they are metabolically stripped down. They completely lack the independent metabolic pathways to synthesize their own ATP or essential coenzymes. They literally have to steal energy from their host. This makes them incredibly difficult to culture in a lab and exceptionally tricky to treat in a clinical setting, mainly because attacking the bacteria often means you have to penetrate the patient's own cells. Let's look at the primary offenders here, starting with the genus Rickettsia. These are tiny, gram-negative pleomorphic rods. Now, because they cannot survive out in the environment, they rely entirely on blood-feeding arthropod vectors, ticks, fleas, or lice, to actually breach the human skin barrier. Once they get injected into the circulatory system, they invade and systematically destroy the endothelial cells lining human blood vessels. This profound vascular damage is exactly what leads to severe clinical diseases like Rocky Mountain spotted fever, which is caused by Rickettsia rickettsii, and epidemic typhus, caused by Rickettsia prowazaki. Now, in stark contrast to Rickettsia, we have the genus Chlamydia. These are extremely small, spheroidal bacteria, but they do not utilize arthropod vectors for transmission. No ticks or fleas here. Instead, they rely on direct human-to-human mucous membrane contact. And man, their global clinical footprint is immense. Chlamydia trachomatis isn't just the most common bacterial sexually transmitted infection globally, but in developing nations, it's also a leading cause of severe ocular blindness, a condition known as trachoma. And this brilliantly illustrates exactly how chlamydia pulls off its intracellular hijacking. It uses a biphasic life cycle. It systematically alternates between two distinct structural forms. Step 1 is the elementary body. Think of this little guy as the infectious scout. It's small, it's rigid, highly resistant to environmental stress, but metabolically inactive.
Its sole job in life is to attach to and enter a host cell. Once it actually gets inside the host cytoplasm, it transitions into step 2, the reticulate body. Now, this form is larger, fragile, and highly metabolically active. It replicates extensively, stealing the host's ATP the whole time before condensing back into thousands of new elementary bodies that just burst out to infect neighboring cells.
All right, let's hit section 3, the extreme rule breakers, domain Archaea. So we often toss around the term extremophiles for Archaea, but their ability to survive extreme conditions isn't just some random quirk. It's a direct result of their profound structural divergence. While they are prokaryotic in shape, meaning they lack a true nucleus, their biochemistry is wildly unique. Instead of the standard ester bonds found in bacterial and human cell membranes, Archaea use incredibly strong ether linkages to bind their lipids to glycerol. Instead of straight-chain fatty acids, they utilize long-branched hydrocarbon chains called isoprenoids. And perhaps most importantly for your upcoming exams, they completely lack true peptidoglycan in their cell walls. Instead, they build their walls out of pseudomurine, complex glycoproteins, or dense protein layers known as S layers. These unique building blocks are exactly what allow for such extreme adaptations. Take hyperthermophiles, for example. These guys routinely survive boiling temperatures that would instantly melt a standard bacterium. Like, no problem at all. How? They do this by fusing those ether-linked isoprenoids into a continuous rigid lipid monolayer. So the two halves of the membrane literally cannot peel apart when exposed to intense heat. Couple that monolayer with specialized heat-stable proteins, which are packed with dense hydrophobic cores and internal salt bridges, and you've got a cell that flat-out refuses to undergo thermal unfolding. Then we have the extreme halophiles, out there thriving in hypersaline environments like the Great Salt Lake. Their survival trick? Intracellular salt accumulation. They actively pump massive quantities of potassium ions directly into their cytoplasm until their internal solute concentration perfectly matches the insanely high external salinity. This structural salt baseline is absolutely vital. It prevents deadly osmotic water loss, and it chemically stabilizes their specialized cell wall glycoproteins. Without that salt, those glycoproteins would literally liquefy and fall apart if the environmental salt concentration ever dropped too low. Finally, for this group, we have the Methanogens. These are strictly anaerobic archaea that consume carbon dioxide and hydrogen gas to generate methane gas as a metabolic byproduct. They hold massive ecological relevance, practically driving the global carbon cycle in deep swamp sediments and marine trenches. But hey, they also have clinical and biological relevance for humans too. They actually reside quietly right inside the digestive tracts of ruminant mammals. And yep, even in the human gut. Which brings us to section 4, the 3-domain matrix, transition to Eukarya. This right here is the ultimate structural bridge connecting everything you've learned so far to the upcoming chapter. When you actually sit down and compare domains Bacteria, Archaea, and Eukarya side by side, a truly fascinating paradox emerges. See, Archaea look like bacteria under a microscope, but at a molecular level, they share deep homologies with us, Eukarya. Think about it. Bacteria initiate protein translation with formylmethionine, but Archaea and Eukarya both use unaltered methionine. Bacteria use a pretty simple 4-subunit RNA polymerase, while Archaea utilize a complex multi-subunit RNA polymerase that very closely mirrors our own eukaryotic enzymes. And while Archaea have 70S density ribosomes, just like bacteria, the actual protein subunit sequences of those ribosomes mirror eukaryotic ribosomes. It's kind of wild. Which leaves us with this final, profound puzzle to think about. If Archaea are structurally simple prokaryotes, totally lacking a nucleus or internal organelles, how do these deep, complex, eukaryotic genetic similarities change our view of our own biological evolution? Did the complex eukaryotic cells that make up human bodies actually evolve from an ancient archaeal ancestor that simply learned to encapsulate its DNA? Keep that provocative thought right at the front of your mind. You are definitely going to need it, because in the very next chapter, we are leaving the prokaryotes behind and diving headfirst into the incredibly complex world of eukaryotic microorganisms. See you there.